Hello, everyone. My name is Vincent Hicks, and I'm a bio major on the pre-med track here at Oglethorpe University. I'm a junior this year, and I chose this topic because coral reefs are very important to our ecosystem and our economy, but they're in a lot of trouble. And pretty much all of this trouble has been caused by us humans. So my goal with this presentation was to raise awareness and bring a little more attention to this problem, since us, the humans, we have to be the fix. First, I'm going to get into the importance of the coral reefs and some plastic pollution issues that the ocean is currently facing. Then I'm going to introduce the study for today and the goals of the study, how they collected their data, the results of the data, the conclusions from the data, and the take-home points for today. So first, I'm going to explain how coral reefs are beneficial to our ecosystem and our economy. And then I'm going to explain a few of the issues that they're experiencing and a few surprising facts that you may or may not have given much thought. And then I'm going to differentiate between micro and macroplastics and the issues that they're causing in the oceans. So first off, we have a global distribution map of the corals in the world. As you can see, as we get closer to the equator, the density increases, which means that they're just pretty favorable towards warm weather rather than cold weather. They're the most dense around Australia over here. As you can see, it's pretty much black. But over here in the States, we have a few, but not many. Just pretty much in Florida, in South Florida, and a little on the East Coast. So first off, coral reefs are very important because they act as breeding grounds and just housing pretty much for invertebrates and fish. So invertebrates can include maybe a squid or a sea snail, but and fish, obviously just fish with bones. But because of the tight space, it just makes it pretty hard for predators to eat the fish or their eggs or their babies after they've hatched. Something else that coral reefs do is they reduce the wave energy. So the waves hit the shore with a lot of force normally, but they kind of act as a barrier to kind of soften the blow. And something else that they do is they filter the sediment that would rather end up on shore. So if you look at beaches with very, very fine sand, it's likely that they have a really healthy coral reef ecosystem, just because they act as filters. So if you go to a beach that really doesn't have many corals in its in the area, then it's more likely that you're going to have coarse sand with rocks and shells and stuff in it. Ecotourism is something that brings in a lot of money for the economy. So like in Australia, how I was mentioning before, the corals are the most dense over there. So the Great Barrier Reef is a very, very big tourist attraction that brings in a lot of money for the economy. Something else that people don't even know is that medication is being produced with coral. Some of the current treatments we have are to cure, not cure, but to treat cancer, arthritis, human bacterial infections, Alzheimer's, heart disease, and there's even more than that. So for here, they have a density map that they used MODIS imagery to capture. So MODIS stands for Moderate Resolution Imaging Spectroradiometer. And what it does is it helps scientists determine the water vapor in the atmosphere and predict climate with that. But in this case, the researchers set it up to detect chlorophyll from cyanobacteria blooms in the Florida Bay. So like I was saying, we don't have many corals in Florida, just in the States and as a total. But as you can see over time, they've just vanished or they've dissipated. As you can see, the density in 2006 to 2007, or 2005 to 2007, you can see they're really dense and there's a lot of corals here. And as you can see, recently, they've thinned out a lot. So you can see that the decrease is evident and you can see it. But the cyanobacteria that I was mentioning is called Synechococcus, and it's not necessarily bad for the corals, but it's just an association. So whenever there's corals, they have this bacteria, just like how humans have good and bad bacteria. Not all of it's harmful. So they just set up the chlorophyll with the MODIS satellite, and obviously the more chlorophyll there are, the more Synechococcus cyanobacteria blooms there are, which means the more corals there are. So these are microplastics. Microplastics are pretty much just defined as any plastic that's under the size of 5 millimeters. And macroplastics are just essentially everything else, everything that's bigger than that. The most common examples of macroplastics that are in the ocean today are just single-use plastics like this. So plastic bags, plastic water bottles, cigarette butts, straws from fast food restaurants and silverware, plastic silverware. So a big problem with this actually though is that turtles and plastic bags do not mix well. Turtles often, very often mistake plastic bags for jellyfish since they kind of look similar and they float freely like this. So they're consuming a lot of plastic and killing themselves or making themselves sick without realizing it. And it's our fault. It's our fault. A fish didn't use this bag. We did. Um, Here we have another example of just plastic that ended up harming a fish. So if a fish as a juvenile, as a baby, gets wound up in plastic, it doesn't just snap the plastic as it grows. It's going to grow around the plastic, as you can see here, this little indentation that's been caused in the fish. And this is actually a Powerade wrapper that this guy found and removed off the fish. But yeah, it's just a piece of plastic that caused this. So if this happened to maybe tangle around its neck when it got young and when it was young and got caught, it could have stopped it from eating by closing up this feeding tube here. Or if it got caught up here or down here, then it wouldn't be able to swim, or that means it couldn't hunt or evade any predators. So even if it can evade predators, it gets eaten. It's plastic. It's just then in another fish. So some of our plastic pollution issues is that harm to fish is also harm to us. We eat fish, and like I said before, the fish that we eat has been consuming plastic. So because of our bad garbage disposal habits, we're introducing these materials into our own digestive systems without even realizing it. Another problem is that these plastics are found in both surface and deep water, so all aquatic life is at risk. Even in the example of coral, there are different species of coral that live, very close to the surface to where you can see from the sky or see from a beach. But there are also species of coral that only live in deep areas of the ocean, like our species today, Lophelia pertussa. But last but not least, they harm the coral reef barrier, and we're about to get into exactly how that happens. A few eye-opening facts for you guys is that every 1 in 3 fish captured for human consumption contains plastic. Just think about that. Microplastics, and macroplastics, they also contain aquatic bacteria, which can have a range of health effects on fish and corals. I'm not going to get into specifics today about, how bacteria affects corals rather than plastic, but a lot of people don't know that fish are kind of like humans in the fact that they can get diseases too. They can get sick too, just like humans can. So these diseases are making fish sick and they're dying from little fish diseases. Another good point is that corals are alive, and a lot of people don't realize this. Corals are not just rocks. They're not even just plants even. We're going to get into exactly how they function in a second, but they they feed. They're an actual animal. They eat, they essentially hunt. So corals are alive. So we need to fix that. We need to fix the problems that we're causing them because, like I said, they're beneficial to us. Here we have a picture of the Great Pacific Garbage Patch. Essentially, it's exactly what it sounds like. A big collection of garbage in the North Pacific Ocean that's been brought together by these currents over here. So these currents are just natural currents that just go through the ocean, and it's just been making this trash travel. This is a picture of the subtropical convergence zone. So you can see it's just this line, and on the other side, it's just an even bigger pile of trash. So something crazy about this is the fact that just in size, it's twice the size of the state of Texas. And Texas is huge. It's twice the size of Texas. And it's also 3 times the size of France. So this is just a big floating patch of trash in the ocean. We got to fix this. Here we have Lophelia pertussa. So this is our species of interest today. This is what our researchers study. and they're a deep sea species. And as you can see, this is a nubbin. This kind of skeletal structure of it is called the nubbin, and these are polyps. So this happens to be a species that was um, actively feeding, which is why you can see these polyps protrude from the nubbins, and these feeders are what catch food. So when they're inactive, however, they go back in and they're just, branch looking. Um, like over here, if you look over here, there aren't any polyps coming out of the nubbin. But this is a pretty close-up picture, so you can see how they function. But this is what they look like in their, natural state, just when you would see one in the colony. They're pretty just, just branch-like, stick-like structures. And they are found in the Mediterranean Sea, deep sea. So the researchers, they went over 500 meters to collect these. So down there, there's no light and it's very cold. So something to keep in mind. For our study, we have macro and microplastics affect cold water corals growth, feeding, and behavior. And for the remainder of this presentation, I'm going to be referring to behavior as activity, but it means the same thing. The growth is obviously how much is growing. The feeding rate is how much it's catching when it feeds. And the behavior or activity is how often those nubbins, those polyps are protruded from the nubbins uh, trying to feed. So first I'm going to give you guys an introduction, and then we're going to get into the goals of the study, the methods of how they collected their data, the results of our study, and our conclusions. So here we have our 10 authors of this study, of this paper, and they worked very hard on this. And they got it published in Nature Journal as a scientific report in 2018. Nature Journal is a very, very prevalent journal for science-related studies. So the goal of our study was to determine if microplastics or macroplastics or both affect the feeding rate, growth rate, and activity of the species Lophelia pertussa. If so, how? Would it make it increase or would it make it decrease? It's hypothesized, however, that both the microplastics and the macroplastics reduce all three of these values. So the feeding rate, the growth rate, and the activity is all hypothesized to drop and lower than the control value, the control species. So for our methods, their goal pretty much for the method was to imitate the natural habitat of Lophelia pertussa as much as possible. So like I said, they're from the Mediterranean Sea and they're deep water, which means that very they're very far, which means that there's no light, and it's also very cold just since there's no light. So that's just something also to keep in mind as we go through these. For the first thing they did was they treated the plastic. They treated the micro and macroplastics that they were going to use to introduce to corals. And what they did was they sat them for 2 months in some filtered seawater. And this was to allow all of the aquatic bacteria to colonize on it before they introduced it to the corals. The reason that they filtered the seawater and kept it circulating is just to, replicate that it's still in the ocean. It wouldn't be using the same patch of water. It would be water flowing by it. So they kept the water circulating and filtered, and they left it in for 2 months to let the bacteria colonize on it. For our coral sampling, like I said, it's from the Mediterranean Sea. They went 540 meters deep to collect them. When they got them and brought them back to the lab, they were transported in a tank that had no lights and no windows or anything. So it was completely dark, and it was held at 13 degrees Celsius. That's the temperature that they're used to and what they live in at the bottom of the ocean. And like I said before, they use filtered and circulated water just to imitate that it's still in its natural habitat, receiving fresh water and fresh oxygen. So for our data collection, they first went over the first hour. They looked at the growth rate and feeding rate and the capture rate, and the activity, the capture rate, and the growth rate over the first hour, 7 days, 20 days, and 48 days. And for the capture rate, they use the species Artemia salina, which is just snow plankton. It's a really, really small species, sort of like brine shrimp, if you know what those are. But for the capture rate, what they did was they just released a number of snow plankton, they counted, and then they counted afterwards to see how many that the corals were able to catch. For the activity, they used night vision cameras at two different angles, a lateral and an aerial view, to observe all of the angles of the polyps and the nubbins. And they just observed how much time they spent outside of the nubbins. So how often they were out trying to feed and for how long. For the growth rate, they took identical sized nubbins of each coral. So for the control, for the microplastic and macroplastic, and for the control, they just didn't introduce any plastics to it, but they also kept it in a tank in complete darkness at 13 degrees Celsius. So they stained three identical size nubbins on each of the different ones, the control, micro, and macro. And then they did that at the beginning. So after each period, so after 7 days, 20 days, and 48 days, they measured the new growth from that stained portion. So they just used the stain as a baseline and they compared all of them afterwards. And for here, they didn't really have any pictures of their experimental setup in the study, in the paper, but this is something similar to what they use to introduce the plastics to the coral and to keep them there. This is called a flume. And what it does is it just keeps the water circulating. So like I said, they need to keep it circulating and filtered to, kind of imitate how it would be in the real ocean, how the Lophelia pertussa would receive water and oxygen in the ocean for real. So down here, this big pipe is water comes through here, comes up and, pushes out and it goes back. So it's just a cycle that keeps it moving. So for our results, what happened? Did the capture rate go up, activity go up, growth rate, what happened? And like I said before, the control was just no plastics at all, no micro or macroplastics. In situ conditions just means that they didn't sample it at all. They didn't take it into the lab. They just left it in its natural environment and they measured from there. And then obviously the micro macroplastics, they introduced microplastics or macroplastics. So here for our capture rate, this was observed over the first hour. So this is 20 minutes, 40 minutes, and 60 minutes. And in the black, we have our microplastics. In our white, we have macroplastics. So over 20 minutes, you can see it significantly decrease, especially in the macroplastics, the capture rate. And this, oh, my bad. By the way, the dotted line is the control. So that's what it's being compared to. So over 20, 40, and 60 minutes, you can see they're all pretty much under the control value, especially except for the microplastics. For 60 minutes, it seemed to kind of leveled itself out a bit. Now, to continue the capture rates, microplastics and macroplastics for 7, 20, and 47 days. For microplastics, you can see that compared to the control, the capture rate decreased on both 7 and 20 days, but not 47 days. There was a slight increase. And if you look at macroplastics for capture rate, there was a strong decrease all across the board for 7, 20, and 47 days. So if we look at the activity, however, compared to the control, for microplastics in 7, 20, and 47 days, they were all negative. They were all under the control, so they all dropped. But for macroplastics, however, the 7 days after a week, it dropped, but after 20 and 47 days, it managed to increase. So there was a fluctuation there. Then for our growth rate, here we have our in situ condition, which, like I mentioned before, they left this in the Mediterranean Sea where it was at, and they just measured it from there. And for our control value, like I said, they just had it in the tank, complete darkness at 13 degrees Celsius. And microplastics, macroplastics, you can see that they were significantly lower than the control values in the in situ conditions. So you can see this is obviously evidence that plastics are stopping the rate at which corals can grow. And by the way, this was taken after 69 days. The other baseline was 47, but they took this one a little bit after. So for our conclusions, what happened? Why did the capture rate decrease? Why did the activity fluctuate? Why did the growth rate decline? Let's get into that. So for the capture rate decline, it's pretty much as simple as this. Plastics are just obstacles. If something is covering it, how is it going to be able to capture food? So as you can see, this is a picture of a plastic bag, and this is Lophelia pertussa. And see, this plastic bag has had bad effects on this plant because this brown portion is where it's died. So this piece is still alive. It may or may not have much time left, but part of the other colony has died. And part of the reason could be because of these plastics. The activity fluctuation, why would it increase? That would be because of scarcity compensation. So if there's less food, means the polyps will want to come out more to catch more since it doesn't come by as often. So if there's less, I'm just, going to stay out more to increase my chances of catching something. But why would it decrease? Like we saw, we had that fluctuation. It went up and down. So it could decrease just because there's just not much energy since it's not much food it's eating. Less energy, just less food it's eating, less energy, it can't continue its natural processes the same. And for our growth rate decline, similar to our activity, it's just that there was less food that it was eating, so there was less energy that it was producing to be able to continue its natural functions. So to revisit our hypothesis that both microplastics and macroplastics reduce all three of these values compared to the control. How do? We right or wrong? So the hypothesis was proven to be correct for the most part, as you can see. The growth rate did what we expected. It dropped. The feeding rate, the capture rate also dropped as we expected, but there was a fluctuation of activity. So for the most part, we were spot on with the hypothesis, but we're a little off. So a few take-home points for you guys is that plastic is the #1 pollutant in oceans. It's the #1. It's worse than runoff waste from different factories or runoff waste from fertilizers from farms. It's worse than scrap metals. It's worse than oil. Plastic is the #1 pollutant. So we need to do a better job of how we get rid of our trash. We need to make sure we're using more reusable objects and recycling when we can, even though it's not 1-for-1. Recycling definitely does help instead of sending everything to a landfill. Second point is that corals are in big trouble, like I've shown you. They have a lot of problems with plastic. They're obviously, like the study showed you, they're not growing as fast. They're not catching as much food. So corals are in big trouble. And we need to fix this since, like I said, there are a lot of benefits that we receive from corals, like from the economy, from tourism, from ecotourism, and also just from our beaches. They reduce a lot of sediment and to help keep the beaches looking nice. And like I said, to give fish and invertebrates a good safe space to reproduce. So third point is that this whole presentation was proof that plastic pollution is a real health issue for aquatic life. It's not just, it's not just a rumor, it's not a myth. These are facts that I've proven, that these researchers have proven that plastic pollution is a real health issue. So these are my sources, and I hope you all take something home with you today and you learned something.